If you look at blood cells under the microscope, this is a cell you don't want to miss. Here is the blood cell we looked at for blood smear quiz number 4. When you're learning to read blood smears, the best thing to do is be systematic. So first thing we're going to look at is size. Most of the time, you're going to estimate size by comparing it to a reference cell. In this case, we can compare it to the reference red blood cells that are nearby, and if we can look elsewhere on the slide for a lymphocyte, a normal appearing lymphocyte is also a good reference cell for these. So this cell is quite large compared to the surrounding red blood cells. The next thing that we look at is going to be the nucleus to cytoplasm ratio. In this cell, it is extremely high. That means the nucleus is taking up a majority of the space inside that cell, and we only see a little bit of cytoplasm. Next thing to look at is the nucleus. And in this case, we can see clear nucleoli, which is an important thing to look for because it tells you about how far left that cell is in the development cycle. We also want to look at the chromatin and how open that is in the nucleus. That's that dark purple stuff. And then finally, we're going to look at the color of the cytoplasm, in this case, very light blue, and the presence or absence of any granules. So in the case of this cell, it is extremely large compared to the reference RBCs. You can fit 3, probably 4 red blood cells inside of this cell. This is a very large cell. The nucleus to cytoplasm ratio is very high. We see mostly nucleus. There are nucleoli. There's sort of open chromatin, and the cytoplasm, maybe there's a couple of little granules in there, but not too many. This is a blast. So if you see this in a peripheral blood smear, this is an acute leukemia until proven otherwise. Now, we can see peripheral circulating blasts in other situations. Probably the most common one is going to be a primary myelofibrosis, but any condition that leads to extramedullary hematopoiesis, which means that there's something going on inside the bone marrow, and so the body cannot make adequate cells in the bone marrow, and so the body starts to produce blood in other organs, usually the liver. So when we have extramedullary hematopoiesis, there isn't that bone marrow structure for the young cells to hang out where they usually hang out inside the bone marrow, right? That's usually where blasts hang out. But if blood is being made in an extramedullary space, those young cells can be released into circulation. So not every circulating blast means that there's an acute leukemia, but you got to be able to identify these cells and urgently make sure it's not an acute leukemia. Follow for more. I'm going to do a whole series on systematically how to read a blood smear with examples.